To stay on schedule, we'll switch to the last talk of this mini symposium, which will be given by Marcel Hörning. He's a scientist at the Institute for Biomaterials and Biomolecular Systems at Stuttgart University. And I see he's already switched on his video and probably ready to share his screen. So Marcel, the stage is yours.
Yes, thank you. I will share. Okay.
Oh yeah, and he's going to tell us about complex lipid dynamics on entire 3D cell membranes. Size and shape matter, just so I've read all the titles and everyone is equal.
Okay, thanks a lot. So yeah, so that's a joint work of Thorsten Bullmann from Leipzig University in Germany and Tatsushi Bata in Leiden and Kobe, Japan. And I will speak about Dictyostelium cells today. So I got introduced like that they're quite fancy cells. They're chemotactic motile cells to study pathological activities such as allergic inflammation, cancer, metastasis. And most papers you see are basically based on single confocal planes where they observe like PIP3 and PIP10 and basically studying also the migration dynamics and signaling pathways throughout the actin polymerization. So even you use an actin depolymerization inhibitor, you still see here in green, well, here in green, quite complex dynamics of PIP3 dynamics on the membrane. And here you see also propagating waves. That is a, oops, sorry. The adhesional part of the membrane directly on the bottom of the dish, and you'll see phase waves like spiral waves and propagating waves. So, but when I was introduced to the system, I was thinking, well, everybody studied this in 2D, and my challenge was a bit to visualize the entire dynamics on the entire membrane, which is not just important for Dictyostelium cells, but the general problem. Well, Dictyostelium is a nice system to start with. And here, from the physics point of view, we're a bit interested, okay, how does that shape and size of that confined 3D system, the membrane, mediates the pattern formation? So here we started with a simple system. We simply used an actin polymerization inhibitor, had these round cells where the PIP3 here in green, always now to see, happily propagates around. So the main aim of this study was basically doing spin confocal microscopy, initially at least, and generate an analytic system or like that can generate as a 3D perspective of the entire cells in time. So what we did, I tried to summarize rather short. We did an integrative analysis of using triangular grids, and where every tetrahedra of the grid is basically analyzed independently to select the membrane location within that cell. And the size of the grid is iteratively adjusted so that the membrane locations have, membrane parts have the same area of about 0.3 square microns. So like that, we were able basically to extract the cell shape. I remind you that is actin depolymerization. So there's no actin. That means we have basically a collapsed cell that is on the bottom. You watch here from the bottom of the cell, a bit 30° around. And what you can do now is basically you simply need to map that signaling. And what you get is something like that. So here's it's a raw signal, a snapshot of a Dicty cell. Green means high GFP. That means you have a lot of PIP3. And red means basically there's almost or no PIP3. And since we are living still in a two-dimensional world, at least scientifically, more or less, we need to project the cell, so otherwise we cannot see on the back of the cell. So here I use a Mollweiter projection that is an area conserved projection. Well, you're used to see the Earth, that's the Earth, so you don't see just Europe and Africa. Now you can see also Japan and the Asian countries. Well, the same is for that cell. So with that, we have basically made a base, and but we were interested in tracking these domains. And for this, we use spherical harmonic analysis in order to simplify the rather complex patterns. So we used this up to order of 3, and basically reproduce these original raw data to a kind of simplified version. And with that, we can now track by simple thresholding the domain locations, and we traced up to 2 peaks at the time. Well, because there were almost no 3 peaks at the time because the cells are simply small. And with that, to keep it short, we could analyze the entire dynamics. You see here 30 minutes of observation, every snapshot, every 10 seconds. And what we can do now is basically from getting all that information, which we can extract now, like not just the signaling, such as speed, distance of these domains, peaks, and the activity itself, we have also clear and precise topological information of the cells, such as volume, adhesion area, adhesion angle, and so on. So there we had a lot of data, and we had a lot of results to show you a few. Basically, you see that these cells, the signaling basically is self-mediating, so that means as bigger the cell, as longer the distance of these peaks. In case you observe 2 peaks at a time, you can see they try to kind of repel each other, so you observe them every time opposite, on the opposite side of the cell. The same like we found that these waves follow the longest pathway of the membrane, on the membrane, which is basically due to the asymmetry of the cell along the circumferences. I mean, equatorial level, if you want. And you find basically a strong correlation between the membrane ratio, that means aspect ratio between adhesion membrane part and the entire. And this one is strongest for small cells. So here, basically, you see circumferences, small cells, and you see the red data points here shows here a really strong correlation between membrane ratio and the directionality of the waves, of the domain speeds along the circumferences. Additionally, we could also extract restitution-like properties, as you can find also for hard tissue, other excitable systems. Here, we have an enclosed, we have a system where we can take the circumferences as a reference point, and you can see as smaller the cell, so as slower the velocity, and because you have a kind of wave-wave interaction, and that inhibits wave speed, similar as if you would pace the waves with a high frequency. So what we can do, so we could derive that superly difficult equation. We could say that the dynamics is equal a function of the topology of geometrical confinement and the excitability. So we could observe the whole zoo of cells. So you can see if the cells are really small, mainly, so you don't see any waves. If you have slightly larger cells, then they start to produce that kind of spot waves. Here you see, again, these 30 minutes of spot waves. You see how they breathe in and out. And as larger the cells and other properties such as excitability, you can see that more and more you get the formation of waves until you get really rapid propagating waves along the circumferences, as most papers like to show because it looks most fancy. But here we have the advantage. Now we can observe every cell, all kind of cells, also the cells that are not that active or show different patterns. And we observed also cells where these waves actually propagate up and downward rather than on the circumferences. So with all that information, we came up with simulations using the scheme proposed by Alai et al. In 2010, which is quite complex. And we used the same now 3D environment as for the experiment. So these kind of 3-dimensional grids. And we used rather realistic values that are mainly extracted from the literature. And that is based on the Leap method. So it's a stochastic simulation following the Gillespie algorithms. And what we can do, well, it's no surprise, we can recover this kind of dynamics. But when we look a bit more in the statistical way, a bit closer, so we simulated about 1,000 simulations by varying morphology such as aspect ratio and size. We could recover the restitution properties. Here you see the velocity pairs along the longitudinal and latitudes. And you can see, depending on the aspect ratio, they behave differently. If you quantify that and compare with the experiments, so here the black dots are the experimental data, and here the red one means, I mean, at 0 means it's a perfect round cell. You see experiment, that's what you expect. Well, there is no asymmetry in the cell shape, so aspect ratio of the velocity should be 1, but the experimental data shows difference. So what we can actually recover is this kind of shape or geometrical defined control of aspect ratio, but not as strong. And we didn't know why. And so we wanted to have a look, a bit more close look at our the complicated equation. And so the first lookup was to change and have a look at the diffusion properties of adhesive membrane part and the outer one. But that couldn't solve the problem. So next idea was, okay, maybe there is a problem or there is some influence of the membrane curvature, because it's quite strong at the interfacial area between adhesive part and outer part. So I will show you now some primary results, omitting all the statistics. So basically just nice figures because of lack of time. And so what we did, we used this data, so the raw data in this case, which we mapped on the 3D spheres membrane. We use determinate fluctuation analysis, and that gives you the possibility of single timelines to correlate basically what kind of noise you have. So if you have, if you get a value of about 0.5, you know you have around white noise. If it's 1, it's like pink noise, or 1/f noise. 1.5 is Brownian noise, and if you have no noise at all, you basically get ballistic motion. It's about 2. So here's an example published before by somebody else, and you can see here is the same, basically same trajectory, only the implied noise is much higher. So you get here a lower value. So that we can do also with our data. Again, we have here now raw data. We do not use the spherical harmonic analysis because that wouldn't make much sense. So you see here the front of the cell, the back of the cell, and the entire cell, and mapped on 2D. And here two positions on the membrane that show different behavior. On the bottom, the red line shows your basically noise dominant signal. That means we get also an alpha of about 0.5, so 0.6 white noise. And here is more, here you see a signal where the PIP3 waves basically pass by periodically. And then you get a value of about 1 or higher, which is about pink noise. So in this experiment, we obtained then values between rather 0.5 and 1.5. And what you can do is you can map that on the entire sphere. So here you can see now one example. So 2 examples I show you now are typical examples what we observed. So one is, well, we observed like this pink noise, so 1/f noise above the adhesion line. Okay, that means below that line, that is a membrane which is adhered to the substrate, and the upper one is on top. So what you can see is basically it's like a really sharp transition. Yeah, you can also, if you plot the histogram of that, that means here you have the bottom of the cell, here the top. You can really see it drops down. So there is not much signaling going on. And if you have a look at that example and look at some snapshots and the raw data, you see really, so that's again, this is a map, and this is a turned view from the bottom, 45 degree. And you can see really how this wave is kind of trapped and guided by these adhesion membrane interface. I call that now. So this dashed line, yeah, I call now adhesion membrane interface, AMI. And you see it barely, there's no signal. There still can be signal observed, but these waves do not pass. And contrarily, we have also a lot of examples like that, where we can see actually apparently, oh, there's almost no influence of the AMI. We can see, have a look at the histogram. You see there's most activity actually at the bottom, and as we observe for almost all cells, as higher you go, as less signal you get. And it's the tops are mostly noise dominant. But here again, we look at the snapshots, and you see even here, you have a kind of influence of the AMI on the interface. where the wave kind of stopped. Eventually, this wave propagates around, and you have a feeling that could be look really like a spiral wave that is pinned to the AMI. So we did a lot of statistics and analysis, but I don't show you that today. So I want to show you some other way of viewing that so that you may believe me. Here we adapted phase analysis using Morlet wavelet transformation from Taniguchi et al. That was the example I showed in the very beginning. They did that on the adhesive part of the membrane only. We did it now on the entire system. And what you see is you have basically here a snapshot, and you take one time-dependent PIP3 signal, and you analyze them all independently. You get the amplitudes phase. From that, you can fit the dynamics here. From the amplitude, you can map it onto the phase, and from this, you can reconstruct then, analyzing all single membrane parts, the phase of it. So the advantage now of it is we can really have a look of these phased at each time point. And here it's an idea where you can see really what I showed you just before, where you see this pinning of waves at the AMI, and you see if you consider that red-blue interface as a wave front, if you want so, then you can really see how that's counterclockwise propagating around these single spot. Okay, and here is an idea of what you can see a bit faster. So that's the initial wave front here. That are the pinning sites, and these black dots are basically kind of conduction block, if you want, interfacial lines, and to really see how that propagates around. The AMI is here, and you have here also the 3D representative images. So more complex dynamics can be also observed. Well, it's a bit confusing to watch it at the first time, but here basically you see the initial wave front, if you want so, and that propagates down and then squeezes. So here are the two pinning sides, squeezes itself here through. It slows down really much. Therefore, you have not the perfect capturing of that analysis, but you see then how the wave speeds up again and propagates along that AMI. So the curvature, the AMI here, which is induced to that system, plays a crucial role to guide the waves. So it cannot just pin, but also guide the waves. And well, now I make a big jump, and I skip all the analysis and show you basically one of the final results. Here again, we plot the aspect ratio of the velocities along the longitudinal and so in XY and up and down direction. And here we could by using the weighted fit, so basically these values, the colors are weighted through the to the detrended fluctuation analysis parameter alpha. You could actually recover something which we would expect, that actually, as we saw it in simulation, that the aspect ratio must be 1. The problem is that if you take the unweighted fit, you have much more cells for certain conditions than others, and therefore, especially the boundaries of that cells are not represented well. So to visualize it a bit clearly, if you have large cells, which are here like the reddish ones, for example, yeah, they are mostly large, they have rather, the contact angle here is rather small, so you see the PIP3 domains that actually get influenced by this curvature, but still can propagate through. Contrarily, if you have, for example, in smaller cells, you have a much larger angle here, and that means the curvature, the local curvature on the interface here is higher, stronger. That means that kind of domain dynamics cannot pass, and so you have a stronger guidance along that AMI. So with that, I want to summarize. So again, we could observe the entire cell, and we found that the membrane confinement in geometry defines really the wave dynamics. So that means by observing the whole membrane, we could say that size and shape matters a lot, and we could quantify all cells without selectivity, because we don't need a lot of signal in order to visualize it, because in some cells, and if you just look in a single focal plane on a microscope, you may not see anything, but the breathing is on a different place. or signaling. So additionally, we could get a 3D view on that dynamics and found also dynamics that wasn't observed before. I skipped that here. And also we could at least partly recover stochastic, the simulation, by simulations, the dynamics, and that part which couldn't be covered, basically we could explain through the local membrane curvature at the adhesion interface, where we see pinning of spirals or guidance of these waves, of these PIP3 domains, and found also that the degree of curvature defines really the directionality and gives that increased aspect ratio of the domain velocity ratio. So here I want to show you my favorite graph. It's actually really not important, but I feel that is quite neat. Here you see a histogram of all the cells. We observed about 200 cells, and you see the cell volume, and you can see at what scales we actually work here. So the volume of the cells is in around of a sub-picoliter, which is actually quite small. I was surprised myself. I had to calculate it around 10 times to be sure that's really picoliter. And well, the system is really powerful and certainly also applicable to other systems. And well, another step is also not just look at PIP3, but all the other signaling. And with that, I want to finish. I'm more or less in time. The first part of the study is already published. You can find it here. And the second part about the curvature, where I skipped most of the statistical analysis part, you may find soon. It will be submitted soon about that or a similar title. Yeah, thanks a lot. And if you have any question, you may also contact me. If you have idea how to use that approach from a different perspective, different cell type, you may just contact me too. And if you have question, I'm happy to try to answer. Thank you very much.
Thanks, Marcel, for the nice talk. Thank you. So currently, I don't see, oops. So currently, I don't see any, okay, now we have a question from Shakti. So in the, I'm just reading them now. In the stochastic simulation, you took realizations of the results or just take the single run results? So I guess the question is, in the stochastic simulations, are the results somehow aggregated from multiple runs?
So we have really 1,000 independent simulations. They all start from the scratch. That means we have a homogeneous distribution of PIP3, PIP10, and all the other species, and we just let evolve the system. So, and the only thing which we change really actively is the geometry and the size. Yeah, and from that you saw basically the plots we obtained the average. Mm-hmm. I hope that answers the question.
Yeah, I allowed Shakti to also unmute himself if he wants to, or her. So I hope that answers the question. Hello? He or she just unmuted him or herself, but I can't hear anything. So otherwise, just post another question. One quick question from myself. Is it, how does the curvature actually manifest in the reaction? So the only place it enters is in the diffusion.
So far, I mean, you know, the curvature I could just realize in the experiment. So we didn't put it in the simulation yet.
Okay.
So, you know, we try to find what's the reason. So we started this analysis.
Okay, just
now
from
the next step, which I haven't done yet, is to update the simulations.
Okay.
And actually, yeah, what I tried so far is just changing the diffusion from the upper part of the membrane and the adhesion part, but that cannot capture that difference at all. So even I go to really extreme cases, unless I put it 0 on the bottom, you don't see something like that.
So you just experimentally see that at the interface something happens.
So far, yes. Yeah. So next step will to try to find a realization in simulation. Yeah.
Okay. Great. And thank you very much again.